Basal cell carcinoma, also known as BCC, is a non-melanin skin cancer. First documented in early 1800s and published by Kromfer in 1900, it's skin cancer that typically arises in sun-exposed areas. It's the most common type of cancer in the world and is diagnosed in roughly 2 million annual cases in America. Some of the most common symptoms of BCC, as seen in the images here, are a sore that will not heal due to bleeding, oozing, or crusting over, a pink or red growth that is indented in the center, or, as seen in the middle upper image, a scar that is white, yellow, or waxy that does not have well-defined borders. BCC can affect nearly any individual. There are certain phenotypic features associated with this cancer. Individuals with fair complexions, including red or blonde hair, light-colored eyes, freckles, and those who can be easily sunburned, are most affected by BCC. People with these complexions have almost a double chance of developing this cancer. There are also environmental factors for BCC. Possible linkage to ultraviolet radiation exposure and arsenic exposure and the depletion of the ozone layer has led to increasing amounts of BCC. Syndromes, additionally, like nevoid basal cell carcinoma, can be associated with increased risk of this cancer.
Basal cell carcinoma is thought to arise from interfollicular basal cells or epidermal stem cells. There are a couple of genes commonly associated with basal cell carcinoma. The patch gene is a tumor suppressor gene. It is inactivated in about 30 to 40% of basal cell carcinomas, and about 40% exhibit signature mutations caused by UV rays. This gene is part of the hedgehog pathway, which is important in regulating stem cell activity, and in a cancerous cell, this pathway is activated due to patch gene being inactivated. Additionally, the TP53 gene has many significant mutations and is associated with over half of all types of cancers. This gene is important in regulating the cell cycle, including tumor suppression by transcriptional activity. The hedgehog pathway is crucial in basal cell carcinoma. When this pathway is inactivated, the patch protein binds to and inhibits the smoothen receptor protein. This prevents the buildup of GLI transcription factors and blocks signal transduction as shown on panel A. In panel B on the right, there is abnormal activation of the hedgehog pathway. The sonic hedgehog ligand binds to the patch protein, preventing it from suppressing SMO, which binds to suppressor or fuse, and GLI transcription factors are able to enter the nucleus and transcribe target genes. Vismodegib is a USDA-approved drug to treat adults with metastatic or locally advanced basal cell carcinoma. This is the first drug to use the hedgehog pathway to inhibit cell growth and proliferation of tumors. Vismodegib is an orally ingested drug that inhibits the smoothen receptor protein in the hedgehog pathway. Although it is rare for basal cell carcinomas to metastasize and spread to distant parts of the body, it happens and can be difficult to treat, which is why this treatment is crucial for patients that are not candidates for surgery or radiation.
7 studies using vismodegib were found upon reviewing published literature. The methods, results, and discussions are summarized in the next slides in order to further investigate vismodegib as a treatment for basal cell carcinoma.
Now we look at the Stevie study, which is the largest vismodegib study conducted to date. With 499 patients demonstrating a median age of 72 years of age, each patient received 150 milligram capsules once a day orally for 28 continuous days. An additional 28 continuous days of non-treatment occurred afterward. This cycle repeated for 3 years, and each patient received approximately 36.4 weeks of treatment. A variety of responses ranging from 27% disease stability to 65% of combined partial and complete responses were seen. There were a variety of side effects, including alopecia and muscle spasms as well.
The study included 15 patients who were eligible for surgery and had at least one tumor to be removed surgically. Many of the patients had multiple non-target tumors, totaling 43 observed tumors. 11 of the 15 patients completed treatment. Each patient was given 150 milligrams of vismodegib daily prior to surgery. The vismodegib treatment typically lasts 3 to 6 months to see ideal tumor shrinkage, but one patient received 9 months of treatment prior to surgery because they wanted to see more tumor shrinkage. While taking vismodegib, patients were monitored monthly to measure both target tumors as well as tumors that were not going to be removed surgically. Following vismodegib treatment, 10 of the 11 patients who completed the treatment underwent Mohs surgery and 1 underwent standard surgical excision. During treatment with vismodegib, most of the tumors observed saw shrinkage. All patients experienced grade 1 adverse effects during their treatment with vismodegib. This study involved 15 patients with locally advanced periocular and orbital basal cell carcinoma who were ineligible for surgery. Each patient was given 150 milligrams of vismodegib daily and were monitored monthly. The average treatment time was 13 months and the average follow-up time was 36 months. Of the 15 patients involved, 10 saw a complete response. However, one of these patients did have a recurrence following their complete response. The other 5 patients saw partial responses, but 2 had disease progression following these partial responses. 93% of patients saw adverse effects.
Here we have efficacy and safety of vismodegib in advanced basal cell carcinoma. 30% of patients with metastatic BCCs had a partial response rate, and 73% of those patients had tumor shrinkage. The median response time of these patients was 7.6 months. Additionally, 43% of patients with locally advanced BCCs and 21% of those had a complete response with no residual BCC present. Biopsy samples from 34 of the 63 patients showed no residual basal cell carcinoma. On this slide on the right, you'll see a figure from the study that shows results of treatment throughout a 20-week course compared to the baseline images. Here we have risk of cutaneous squamous cell carcinoma after treatment of basal cell carcinoma with vismodegib. For patients treated with vismodegib, 5% developed SCC after 1 year of beginning treatment, while for patients treated with standard therapies for BCC, 4% developed SCC after 1 year of beginning treatment. These values do not statistically show a higher risk of squamous cell carcinoma development after treatment with vismodegib. The image on the slide shows examples of SCC and represent what this study investigated the risks of developing. Here we have vismodegib-resistant mutations are not selected in multifocal relapse of locally advanced basal cell carcinoma after vismodegib discontinuation. Each patient had multifocal relapses after stopping treatment, which can be seen in Figure 3. The results show the same initial mutations typically shown in BCCs were present in the relapses. No further mutations developed, but the BCC was able to grow. Although treatment with vismodegib appeared successful, patients had relapsed. 2 of the patients were again treated with vismodegib and achieved a complete response. This study is crucial in identifying the condition of a patient post-treatment.
In these figures, you can see vismodegib as the green inhibitor on the left and purple cylinder on the right. It functioned as an inhibitor of the smoothing receptor protein. The hedgehog pathway is activated due to the inactivation and inhibition of the patch protein by the sonic hedgehog ligand. Vismodegib blocks the pathway by binding to SMO, preventing GLI transcription factors from entering the nucleus and transcribing target genes.
The development of any drug or treatment option for cancer or any other disease must start with a basic understanding of cellular mechanisms. Knowledge of the hedgehog pathway and subsequent abnormal cell growth has allowed scientists to take advantage of the pathway and halt the spread of cancerous cells. This ability to treat advanced cases of basal cell carcinoma with non-invasive techniques is crucial for patient health. Patients are not always eligible for surgery for a variety of reasons. However, patients with metastatic or locally advanced BCC have more treatment options available due to the success of vismodegib. Thankfully, vismodegib is a better and more efficient option for treatment than previously available.